Turin, Italy, May 28, 1898. A photographer named Secondo Pia is standing in a darkroom watching a photographic glass plate develop in a chemical bath. He is the first person ever permitted to photograph the Shroud of Turin under proper lighting conditions, and he has spent 2 evenings setting up his equipment, negotiating the angles, adjusting the exposure times. He is an experienced photographer. He knows exactly what a developing negative is supposed to look like. The lights become darks, the darks become lights, the tones reverse. It is a mechanical and entirely predictable process. And then the image on the plate resolves, and Secondo Pia almost drops it. What he is looking at is not the expected reversal of the faint, brownish, difficult-to-read impression visible on the cloth itself. What he is looking at is a fully resolved, high-contrast, three-dimensional photograph of a human face. A face with closed eyes, with a beard, with visible wounds on the forehead, the wrists, the side. A face so clear and so anatomically precise that it looks not like a negative image of a body wrapped in linen, but like a modern photographic portrait of a man at rest. Looking at it, Pia later wrote, his hands trembled. He was afraid he would drop the plate. He was afraid he was hallucinating. He was not. What Secondo Pia had just discovered was the photographic negative of the Turin Shroud, which means that the image on the cloth itself was a positive image all along, a perfect photographic positive created by some mechanism that nobody in the 19th century had ever conceived of, and that nobody in the 21st century has been able to fully explain. Whatever produced the image on the Shroud of Turin produced something that behaves, under the chemistry of photographic reversal, exactly like the negative of a real photograph, which means that whatever it is, it encodes depth and light and shadow information in the precise way that a real photographic image does. It means that a 14th century forgery, the explanation most secular scholars default to, based on the radiocarbon dating of the cloth, would require a medieval forger to have produced, by whatever technique they used, a perfect photographic negative 600 years before photography was invented, with a depth-coding precision that modern imaging technology has since confirmed is accurate enough to reconstruct the three-dimensional surface of the human body it appears to depict. You have almost certainly seen the Shroud of Turin before. You have probably seen the face that Secondo Pia discovered on that developing plate in 1898. What you may not know, what most people who have seen the image do not fully understand, is that the debate about what it is has never come close to being settled. And that in October 2015, and then again in August 2026, a team of geneticists from the University of Padua published results of DNA analysis on that cloth that did not end the debate, that moved it, that added a layer of evidence that no other test had produced, that mapped, in the chemical language of genetic code, every hand that has ever touched the Shroud of Turin, every environment it has ever passed through, and at least one specific geographic origin story that the genetic data points toward, with a consistency that nobody who has examined it has been able to dismiss outright. This is what the DNA found. And this is why what it found is more complicated, more specific, and more genuinely interesting than any of the headlines about it have managed to convey. The Shroud of Turin is a length of linen cloth approximately 4.4 meters long and 1.1 meters wide, just over 14 feet by 3.5 feet, which currently rests in a nitrogen-filled, climate-controlled case in the Cathedral of St. John the Baptist in Turin, Italy, accessible for direct viewing only on the rare occasions when the Catholic Church permits its public display. Its surface carries a faint, brownish, bidirectional image showing the front and back of a human body, a man who appears to bear wounds consistent with crucifixion, scourging, and the wearing of a crown of thorns. The image appears only on the outermost fibers of the linen, not penetrating into the cloth, and contains no detectable pigments at the fiber level. It was examined in 1978 by a team of 40 American scientists organized under the acronym STIRP, the Shroud of Turin Research Project, who spent 5 days in direct contact with the cloth, running every non-destructive test available to late 20th century science. Their published conclusions confirmed that the image was not produced by any known artistic technique. They did not propose what had produced it. The most significant scientific result directly bearing on the Shroud's age came in 1988, when the Vatican authorized a small sample of cloth to be taken from one end of the Shroud and divided among 3 independent radiocarbon dating laboratories at Oxford, Zurich, and the University of Arizona. The 3 labs, working independently and blind to each other's results, all dated the sample to approximately the same period, 1260 to 1390 CE, comfortably, within the range of the medieval period, and consistent with the first historically documented appearance of the Shroud in France around 1354. By the standard of radiocarbon dating science, this result is robust. It established what the mainstream scientific consensus has held ever since: the cloth is medieval. Proponents of the shroud's authenticity have argued, in varying degrees of technical specificity, that the sample used for carbon dating was taken from a repaired section of the cloth, a patch added in the medieval period, rather than from the original 1st century linen. The argument has not been resolved to either side's satisfaction. Independent researchers have disputed the claim. The Vatican has not permitted additional sampling for reanalysis. The radiocarbon dating stands as the strongest single scientific argument against the shroud's authenticity. And it stands in direct tension with the image analysis and with every other line of evidence pointing in the other direction. This is the stalemate that existed when a plant genetics and genomics professor named Gianni Barcaccia at the University of Padua decided to try something no one had specifically done before. He vacuumed the shroud. Or more precisely, he obtained access to dust particles that had been vacuumed from the cloth during previous handling, including from 2 specific areas, the section containing the body image and the lateral edge that had been used as the sample point for the 1988 radiocarbon dating. From this dust, Barcaccia's team extracted whatever genetic material had survived: pollen grains, plant cells, human skin cells, everything that had drifted onto the cloth or been shed by anyone who had touched it across the entire span of the shroud's existence. And then they sequenced it. The results, published in scientific reports in October 2015, were dense and layered in the way that genuine scientific findings are when what the data produces does not resolve cleanly into a single headline. The plant DNA told one part of the story, the human DNA told another, and the 2 parts, read together, produced a geographic picture that has not been replicated by any other method applied to the shroud in its entire documented history. On the plant side, the team identified 19 distinct plant taxa, species or genera, in the dust. Several were entirely consistent with a European or Mediterranean cloth that had spent several centuries in France and Italy: European spruce, Mediterranean clovers, ryegrasses, plantainess, chicory. But others were more geographically specific and more difficult to explain through European handling alone. Rare East Asian pear and plum trees had left traces. Plants whose primary distribution falls in the Middle East and the Arabian Peninsula were present. And the analysis also found plant DNA from species that did not arrive in Europe until after 1492, when Columbian exchange began introducing American plant species to the Old World. Traces the T. Barcaccia's team attributed to modern contamination and excluded from the geographic analysis. The post-Columbian contamination was actually useful. It confirmed that the methodology was capable of detecting exactly when material had been added to the cloth, even if it could not definitively date the cloth itself. The human mitochondrial DNA was the finding that stopped people. Mitochondrial DNA, passed from mother to child and accumulating mutations at a known rate, is the primary tool for tracing maternal lineages across populations and determining where a person's ancestors lived. What Barcaccia's team found in the dust was not a single human mitochondrial signature, nor the scattered and random contamination pattern you would expect from a cloth that had spent several centuries in European hands. They found a structured, geographically specific pattern with a clear statistical center. The human haplogroups, the population-level categories that mitochondrial DNA encodes, distributed across the samples in a way that pointed overwhelmingly toward one region. More than 55% of the human DNA signatures came from haplogroups typical of the Near East and Middle East. Less than 6% came from haplogroups characteristic of Western Europe. The rest, a surprising and initially puzzling 38%, came from the Indian subcontinent. And critically, the oldest DNA fragments, identified by their shorter length, DNA breaks down into smaller pieces over time, so shorter fragments are statistically older, came disproportionately from the Indian-associated haplogroups. Barcaccia drew a specific conclusion from this age gradient. If the Indian-associated DNA fragments are older, more degraded, therefore more ancient, than the Near Eastern DNA fragments, it suggests that the cloth itself was in contact with Indian populations before it was in contact with Middle Eastern populations, which is consistent with a specific historical pattern. The production of fine linen in India traded across the ancient world through routes that passed through the Levant and the Eastern Mediterranean, specifically including linen used in the Temple of Jerusalem. Paleographer Ada Grossi noted that historical records document the use of fine Indian linen for the garments of the high priest in Jerusalem, worn during the Yom Kippur rituals. If the shroud was woven from linen that passed through that trading system before ending up in Jerusalem in the 1st century, or before being manufactured into a cloth that was sold in Jerusalem, the DNA gradient from India to the Near East would be exactly what you would expect to find. Among the Near Eastern haplogroups specifically, Barcaccia identified one that he described in a subsequent interview as particularly striking. one of the most abundant human mitochondrial haplogroups found on the shroud, he explained, is still very rare in Western Europe, and it is typical of the Druze community, an ethnic group with origins in Egypt that lives mainly in restricted areas between Syria, Jordan, Lebanon, Israel, and Palestine. This haplogroup is not randomly distributed across the Middle East. It is concentrated in a specific geographic subregion of the Levant that corresponds precisely to the area associated with 1st century Jerusalem, with the communities that surrounded the city in the period to which the shroud's image has traditionally been attributed. This does not prove the shroud is what the Catholic tradition claims it is. The researchers themselves were explicit about this. The haplogroup distribution tells you about everyone who has touched the cloth across its entire history, not about any specific individual. The shroud's documented history in Europe begins in France in 1354, and before that, its history is uncertain. It passed through Constantinople, possibly through Edessa, possibly through other hands that the historical record does not clearly document. Any of those pre-French custodians could have left Near Eastern DNA on the cloth. A medieval French pilgrim who had touched it in a Middle Eastern setting could have transferred those haplogroups. The DNA is consistent with a 1st century Palestinian origin, but it is also consistent with a medieval cloth that passed through the Eastern Mediterranean before ending up in Europe. Then came August 2026, and the second wave of findings from Barkatsch's laboratory, this time working with a different and in some ways more powerful dataset. The 2026 study, published in Scientific Reports on August 4th and summarized by Science daily on the same day, was built on a different sample source. Rather than dust vacuumed from the cloth during routine handling, the 2026 team analyzed official samples that had been collected in 1978 during the STURP examination, samples that had been archived in controlled conditions for nearly 50 years and had never been subjected to the metagenomic approach that modern sequencing technology makes possible. Metagenomics does not target specific genetic sequences. It sequences everything present in a sample, human. Complexity of the material to emerge from the data rather than being filtered by what the researchers expected to find. 3 of the 1978 samples produced high-confidence, complete human DNA profiles. All 3 matched the same individual. That individual was identified, through kinship analysis, as Pierluigi Baima Bologna, the forensic medicine professor who had collected the official STURPy samples in 1978, and who had spent decades subsequently working with shroud material. The match was consistent with Baima Bologna himself, or a first-degree relative. Baima Bologna died in Turin in November 2025, several months before the study was published. His mitochondrial haplogroup, K1A-B1A, was identified repeatedly across the samples. That haplogroup, the researchers noted, is most commonly found in Ashkenazi Jewish populations. The finding was not embarrassing, it was illuminating. It demonstrated with unusual precision and a specific named individual how completely the DNA signature of a person who regularly handles an object can come to dominate the genetic signal recovered from it. If the last person to handle the 1978 samples could be identified by name from the genetic residue he left behind, then every other DNA signature in the same samples, from everyone who had ever touched the cloth or the samples before Baima Bologna, was present at lower concentrations, diluted by time and overprinted by the most recent handler's abundant, well-preserved genetic material. The finding calibrates the entire DNA project, giving it a concrete example of how the contamination chain works and what it means for interpreting the earlier dust sample results. Beyond the Baima Bologna finding, the 2026 metagenomic analysis added several new layers to the biological record of the cloth. The presence of archaea, single-celled microorganisms distinct from both bacteria and conventional eukaryotic cells, was documented in the samples, and specifically halophilic archaea, organisms that thrive in hypersaline environments, the kind of extreme salt concentrations found in places like the Dead Sea and the Jordan Valley. The presence of this specific class of microorganisms on the cloth does not prove the cloth was in proximity to those environments, but it is consistent with that proximity in a way that ordinary European storage environments would not produce, and it adds a specific biological data point to the accumulating geographic picture. Mediterranean red coral DNA was also detected, Corallium rubrum, a species native to the Mediterranean basin, whose presence the researchers attributed to possible transit through or proximity to Mediterranean maritime environments. Plant DNA included wheat, carrots, and various cultivated species, as well as post-Columbian introductions, including maize, bananas, and peanuts, consistent with the cloth's documented presence in Europe from the medieval period onward. Animal DNA included cattle, pigs, chickens, dogs, and cats. The biological residue of environments where domesticated animals were present, entirely consistent with medieval and early modern European storage. The researchers were careful about what their conclusions could and could not support. This study, they stated explicitly, demonstrates the power of modern forensic genomics and proteomics to investigate the biological history of objects. It does not determine the age or authenticity of the shroud. It cannot isolate any DNA that is specifically original to the cloth, as opposed to contamination from the centuries of handling that followed its creation, whenever that was. What it can do is build, layer by layer, an increasingly precise record of every environment the cloth has passed through. A biological autobiography of a piece of linen that has spent an extraordinary span of time in an extraordinary variety of hands. What those layers of evidence add up to is a picture that would be impossible to dismiss as random. The Near Eastern dominance of the human haplogroups, the Druze-associated lineage concentrated in the specific geographic subregion of the Levant, most associated with first century Jerusalem, the Indian linen connection suggested by the oldest DNA fragments, the halophilic archaea consistent with Dead Sea proximity, the Mediterranean coral. Taken individually, each of these findings has an alternative explanation. Medieval transit through the Eastern Mediterranean, a cloth that was handled by people from many backgrounds in or Venice, microbial contamination from a saline storage environment. Taken together, they produce a geographic picture that clusters consistently around the same part of the world, the eastern end of the Mediterranean, the Levant, the specific geography associated with the first century events to which the Shroud's tradition is attributed. That cluster is not proof of authenticity. The radiocarbon dating remains, and it dates the cloth to the medieval period. The image remains, and it remains unexplained by any known natural or artistic process. The DNA remains, and it points toward the Levant, without being able to tell us who left it there, or when. What the cloth contains, after all of this investigation, is not an answer. It is a layered biological record of everywhere it has been and everyone who has touched it, written in a chemical language that took until the 21st century to begin to read. And what that record says, as precisely as it can say anything, is that this cloth has been in contact with the world that produced it, whatever that world was, and whatever that cloth originally was, in ways that no medieval European forgery, created from scratch in France in the mid-14th century, would be expected to leave behind. Secondo Pia's hands trembled when he saw the image appear on his developing plate in 1898. He was not a credulous man. He was a scientist, working with equipment, following a chemical process, expecting a predictable result. What he got was not predictable. What the DNA researchers have been finding since 2015 is not predictable either, not in the direction of proof, not in the direction of easy dismissal, in the direction of a mystery that keeps getting more specific and somehow, therefore, more mysterious with every new technique applied to it. Subscribe, because some questions get bigger the longer you look at them.